Thank you. Thank you so much. I would like to really thank the organizing committee of SSBMT for this kind invitation, and in particular Dr. Ahmed Al-Askar. So this is my financial disclosure. The topics I'm going to go through today, I'm just going to start with a brief story, maybe for the sake of entertainment at the end of the day, and then we'll touch base quickly on the types of cellular therapy, and then I'll spend most of the time talking about the phase 1 study design and focus on the phase 1 design on cellular therapy. So we'll start with a story. It starts with the Jesse was born in Arizona in 1981, and he was born with an inborn error of metabolism that causes liver dysfunction. Jesse was, however, fortunate to have a mild form of the disease, thanks to the germline mosaicism that he's had, and he survived on restricted diet. In 1999, Jesse heard about a gene therapy study that being conducted in University of Pennsylvania, specifically treating inborn error of metabolism. And despite having a mild form of the disease, he envisioned that if he volunteered in this study, he may actually help some people down the road. In 1999, Jesse was 18 years old, and he was injected the adenovirus vector carrying the corrected gene in a phase 1 study. Jesse developed severe immune response and multi-organ failure. He died 4 days after the injection of the adenovirus vector. And of course, this was in the news headlines, as often happens with bad things happening in research institution. The FDA investigated this matter, and they actually faulted UPenn for the conduct of the study. They specifically said that Jesse was not eligible for the study. It was a huge drama at UPenn at this time. More details can be found if you guys look this up. And despite this gene therapy helping a few little kids all over the world, 2 other kids died in France a few years later. So sadly, this euphoria, referring to the initial success, turned to alarm at the end of 2002 when 2 of 10 children died in France with this. And based on this, both FDA in US and France decided to hold gene studies at that time. However, more than a decade later, 2001, Barbara and Edward Netter, having watched their daughter-in-law dying of breast cancer after multiple chemotherapies, decided to start an alliance for cancer gene therapy, basically trying to find something innovative beyond chemotherapy and trying to revive the concept of gene therapy or cell therapy in cancer. In 2008, this alliance granted Carl June $1 million to develop CAR T-cell therapy. And this $1 million was all enough to treat 3 patients, by the way. And there is a long story that took place at that time that I'm not going to go through. But back to where the story started, again in Arizona, but this was talking about Emily Whitehead. She was a child that born in Arizona. And unfortunately, when she was 7 years old, she developed ALL. And this was refractory to treatment, had multiple relapse. And she was advised that hospice is the only treatment or the only way to go for her. There was nothing really. At that time, her parents, out of desperation, heard about the gene therapy at UPenn that opened in the CHOP Hospital in 2012. And she enrolled in a CAR T cell therapy at that time. And despite her suffering of a life-threatening CRS, and she almost died actually, however, she survived. And that's another story worth of another talk. But likely with saving her life, the CAR T therapy, which is a combination of cell and gene therapy, was revived again. And 5 years later, the same product that revived Emily, T-cell, was FDA approved. And you can see here how long does it take from infusion of the from the first infusion of the experimental product in human until it's FDA approved. So that's another product, actually, axi-cell, different than the one that we talked about, and the life cycle here was about 8 years and which is very similar to the T cell as well. So there are multiple cell therapies now available. There are 6 FDA approved CAR T cell therapies, as you can see here, for several indications. And not only CAR T, but there are actually multiple other cell and gene therapy that also FDA approved. So anything in this with cell, it's a cell therapy product. It has a gene in it, and these are all FDA approved, and these are vectors, adenovirus vectors, injected either intravenously or intralesionally to treat different diseases, blood diseases, and non-hematological disorder as well. And there are also myriads of adoptive cell therapy that you can see besides gene therapy, including NK, tumor-infiltrating lymphocytes. They are in development. So how did we get here? One, by realizing the unique nature of the cell and gene therapy and how different they are from the chemotherapy that we have used traditionally to treat cancer. And the other thing, by having the regulatory guidance from the FDA, EMA, and other regulatory organizations making their guidelines publicly available. At least I know about FDA. All of their guidelines are publicly available. And there's just a table comparing the guidelines available for different organizations, including FDA, EMA, and other 2 Asian societies. So phase 1 study design. What is the purpose of the phase 1 clinical study is basically to identify the appropriate safe dose for phase 2 study. The traditional phase 1 trial designs, which has been basically designed for cytotoxic therapies for decades, explicitly assume that toxicity increases with dose levels, and so is efficacy, hence justifying dose escalation. So for phase 1 study, how do you do it, and what do you exactly identify? So traditionally, the phase 1 study has been looking at the maximum tolerated dose, and which is defined as the dose that's associated with inducing dose-limiting toxicity in less than 1/3 of the patient. And less than 1/3 of the patient is arbitrarily defined, and this is what we refer to as target toxicity level, TTL. I'm going to be using this term throughout the presentation. This can be different in different studies, but the reason this was set relatively high here is because we are treating cancer, and that's a deadly disease. So as you can see here, there's just a dose-response relationship that you can see here. You can achieve about 50% response most of the time with a small dose, but you have to have a huge increment in the dose to achieve more than 50% and get closer to 100%. And this is where we start to look for the maximum tolerated dose. And again, this curve is the same thing here. If you pick dose level 3, you can achieve 20% target toxicity, and you achieve about 45% efficacy. But if you go a higher dose level, you can have higher target toxicity level, meaning that 40% of people will have dose-limiting toxicity, but you can achieve higher efficacy. So the question is, do we really have to reach the maximum tolerated dose? What if the pharmacodynamic studies, pharmacokinetic studies show that this is unnecessary? For example, if the pharmacodynamic studies basically looking at target saturation. What if you achieve target saturation before the maximum tolerated dose? What are you going to get with escalating the dose? Same thing for if you're using an oral medicine, and you found out by PICA study that this oral medicine is not absorbed beyond certain dose. So with dose escalation, you will be getting nothing but GI toxicity. What if the MTD is not reached by the proposed dose cohorts? Are you going to keep going forever? This was an interesting study looking at the data of about 700 patients from 24 trials at MD Anderson. And what they looked at here, they looked at the overall survival and actually disease-free survival, which I'm not showing here, comparing the efficacy of low, medium, and high dose used in phase 1 studies. And surprisingly enough, they found that the survival was similar using these doses. And this was for molecularly targeted agent, by the way, not for chemotherapy, of course. So this has just basically implies that reaching the highest or maximum tolerated dose may not be necessary in molecularly targeted agent. Therefore, this terminology is now being developed, the recommended phase 2 dose instead of maximum tolerated dose. The beauty about the recommended phase 2 dose is includes not only toxicity endpoints, but also includes the PK and PD studies. And as a matter of fact, the recommended phase 2 dose endpoint now is required by FDA rather than maximum tolerated dose, and this is for all oncology and biologic products. And then we'll come back to how we're going to get to these dose identification. But how is phase 1 studies done? So traditionally, they've been done by using 3+3 design that we probably most of us know. And this has been used for decades. And again, this is based on using a target toxicity level, which is about 1/3. It's very simple, easy to use by clinician. You still need help from a statistician, but it's easy to understand. But the question, is it the best model? So this is a 3+3 design. It's very simple. You get no toxicity, you escalate, you get one toxicity, you expand, and decide later on. But there are several drawbacks of the 3+3 design. Number one, it's only based on toxicity, because you don't look at anything else. It does not incorporate BD or BK endpoints, which is a big downside now with molecular targeted agents. The other thing, if you specifically use 3+3, you're kind of stuck at the target toxicity level of 33% of 1/3 of the patient. Other thing, it doesn't capture delayed toxicity. So let's hear what is unique about phase 1 studies in cellular therapy. Well, first of all, the context of cellular therapy, you need to understand this could be a novel cell or gene therapy product that's just being used de novo, or a drug or cell product being used in the context of another cell therapy, which is usually stem cell transplantation, such as using post-transplant TKI. So what is unique about this is you can have myriads of unique toxicities with cell therapy that you don't typically see with chemotherapy or even molecular targeted agent, such as GVHD, if you're using allogeneic stem product, graft failure or graft dysfunction, infusion reaction, cytokine release neurotoxicity. These are things that are really unique for cellular and gene therapy. And unfortunately, none of these can be incorporated in the dose-limiting toxicity, which is often said at 33%, because you don't necessarily have to accept a 33% risk of GVHD grade 3 or 4. And same thing for CRS, advanced CRS, because that's going to be really too high. So, and therefore, a new design has been developed for phase 1, which is called model-based designs. That's in contradistinction with the 3+3 called rule-based. So when the model-based phase 1 studies, you can incorporate the toxicity endpoints that we were talking about, and you can actually include other toxicity endpoints, and it choose a different target toxicity level. It doesn't have to be 33%. You can be 33% here and 20% here. And you can also incorporate PD and PK endpoints. Here we talked about the target toxicity level. And the nice thing is with these model-based designs, you can look at the outcome of patients in the study cohorts, even if these cohorts have been passed. Like if you can be in cohort 3 and you're still looking at the outcome of cohort 1, and this will still be informing the decision to escalate the dose. We'll see how this is done. So the commonest one in this context is the continual reassessment method, CRM, and this was developed by two statisticians in Fred Hutch in 1990. Since then, this has been modified a few times. One of the most famous modification is time to event, CRM, developed by these two statisticians in the University of Wisconsin. I believe they are in MD Anderson now. And there are others. Modification done for CRM. So what is CRM? CRM uses what we call Bayesian statistical model to estimate the relationship between the dose and risk of dose-limiting toxicity. The Bayesian approach is a probability theory, very well known among the statisticians, that predicts event based on the prior data. And we're going to see what does it mean. So first of all, we have to start with a starting dose, and we build the model. And of course, this dose is determined based on prior data and also clinical experience, because sometimes it's hard to really decide how we're going to start. We don't have preclinical data for every cell therapy product that we use, because this is actually with the rationale for 3+3. Usually start with 1/10 of the toxic dose that used in preclinical model. But here you don't have to do this. You can actually use your clinical experience and similar data in similar studies. And you can say, hey, I'm going to start with NK dose 1 × 10⁶ based on what we know about data in allogeneic transplant. Something like this. And then you build a model. So this example I'm going to use. It's a simple. The reason I'm using this is because it's really simple and it shows what CRM is. This is a study done using lenalidomide for post-transplant relapse in MDS after allogeneic transplant. This is what the clinical investigator needs to give this statistician to build a model. He needs to tell him what's your target toxicity level. So here they pick 20%. They are not obligated to pick 33%. And they give them the dose level. So this is the LSITD level was fixed, but they pick different lenalidomide dose. And they have to start with, they have a starting dose here, but they have to give the statistician a guess for the maximum tolerated dose because it's going to be the last cohort. And maximum cohort size. So what interesting thing about the CRM is the cohort size is not fixed. You have to have a minimum and a maximum. So the cohort size can be variable. So you can have a cohort of 2 patients, and you can have a cohort of 5 patients, and 3, whatever. But you have to give the determination to statistician to start with, and I'll tell you why in a minute. So the study started here with a dose level 0, was cohort 1. And based on the data that statistician showed with the shared with the clinician, they decided to escalate to level 1 and level 2 and level 3. And here they said, oh, we are not going to escalate further. We are going to expand. And this is the thing. Here, when you expand, you go to another cohort. You don't expand in the same cohort because the cohort has to have a maximum capacity. Remember, this is all statistical modeling, okay? So you expand. And then after they expanded, they said, hey, what do you want to do further? This is the data we have. I'm going to show you this is the next slide. They said, we're going to expand further. And that they keep doing this until they reach it here, and they said, this dose level 3 is the maximum tolerated dose or recommended phase 2 dose, okay? So forget about this writing here. Just look here. So this is where we started. This is the dose level 0, and this was what the statistician showed the clinician. They told him, this is what we have here. And based on this, it turned out that initially it seems like we were overestimating the toxicity of the medicine, because it turned out that at dose level 0, you were far away from the target toxicity level. You still have huge room to go. So imagine, we don't see this yet, okay? We're just looking at this because that's all what you're seeing when you're at dose level 0. Don't be confused by this, because this curve is the prediction. When you look at this curve, it tells you this is the predicted dose limiting toxicity. It basically tells you when you look at this, the predicted dose limiting toxicity will be reached at dose level 1, but you are at dose level 0. So you have room to go, okay? So you went already to 1, and when you went already to dose level 1, this is what the statistician showed the clinician. And they said, oh, the curve really went down. This means that you really have more room to go. Okay, you can actually give dose level 2 and you're still far away, or still away from the dose limiting toxicity to escalate to dose level 2, okay? And when they looked here, level 2 is here actually, it's overlapping, okay? So when they went here, it basically went even further down. So that tells them that you can actually go further to dose level 3, and they did. They went to dose level 3, it went further down. This means that you can go further, but here was the catch. Dose level 3 was basically the maximum approved dose for lenalidomide. Where it's okay. Dose level 3 was 25 milligrams of lenalidomide. So if you really want to escalate, you're taking a risk because you're going above the FDA approved dose for lenalidomide for multiple myeloma, because the next step would be 30 milligrams, which is actually not even built up in their initial cohort. So they didn't want to do this. They decided we're going to go cohort 4 is going to be an expansion. But there's the beauty of this. When they went to cohort 5, the curve started to go up. Okay, and the same thing for 7 and 8, or 6 and 7, basically they are here. So what happens here is basically with further expansion, these curves changed, they went up. So eventually, they decided that level 3 was the maximum tolerated dose that you couldn't exceed, because this is what they get them really very close to the target toxicity level. I hope this is clear, but the beauty of this is they didn't have to exceed the dose limiting, they didn't have to exceed the MTD to say, oh, we have exceeded the MTD like you would often find in this 3+3 design. But instead, they can actually keep expanding until they find a beautiful spot. So the CRM, it has pros and cons. It's flexible, it's safe, incorporates several toxicity endpoints. As I said, you can have a model for the dose-limiting toxicity, and another one for GVHG, another one for cytokine release. You can do whatever you want. You can actually look at the BD case studies. This was not part of what I showed, but you can actually have this and look at this, because this data will be in front of you before you make any further expansion. If you do a PD study and found that you have a target saturation, you can stop here and say, hey, enough. I'm not going to escalate further because I'm not expecting clinical benefit. And I said there's a flexible cohort size. I'm going to give you examples. Let's say you have a study, and the toxicity assessment, you have to wait for 50 days. And you are in cohort number 3. And you have one patient just got the treatment yesterday, and we have to wait for 50 days before you enroll the second subject, because that's how you assess the toxicity. But you have another subject who is ready to enroll. Okay, so traditionally we turn these people down and tell them we don't have a room for the study. But in this, if you're using this model, you can actually enroll them in the cohort before the cohort that you are in, because if these prior cohorts haven't reached their maximum capacity. And this will help any enrollment in the prior cohorts. It helps in this curve modeling, and it still informs the study. And as I said, you will unlikely exceed the maximum tolerated dose with this model. The problem is it's relatively complex and demanding, and requires continuous engagement from a statistician and active engagement by a study team. And these are examples of software available that can use CRM. Luckily enough, R is one of them. These are free. Some of them are free softwares. These are again examples of the CRM studies done expressed in different ways. So here these are the number of cohort, and the red one will be the maximum tolerated dose. And again, here this will be the curve that the statistician will show with the physician, and you can fine-tune the target toxicity level the way you wish. You want to have 33%, you have less or more, and this will basically, it will tell you if you are here, this is what you're likely going to be your best dose to use. And this again also another example of representing CRM, but that's different here. Instead of having cohort here, you have individual patients with individual dosing. But the bottom line, this is what eventually you look at, and this will give you the prediction of the recommended dose or the maximum tolerated dose that's close to the target toxicity level that you can set. So as I said, FDA now mandates using a recommended phase 2 dose for submission. And as a matter of fact, this was a study done like 5-6 years ago that showed that the use of the recommended phase 2 dose endpoint was associated with more FDA approval. And actually also this recommended phase 2 dose was more concordant with the FDA approved dose. So in essence, we have two basic designs for phase 1 studies. One is a rule-based design, 3+3, which is historical, ideal for chemotherapies. But the one that we should really probably follow now, in particular with cellular gene therapy and molecular targeted therapy, is the continual reassessment method and its modifications. In the last minute, I'm just going to share with you my personal experience with phase 1 studies. This was years ago when I was at UAB. We devised a study to use to infuse alpha-beta depleted T-cell product after haploidentical transplantation. After haploidentical transplantation, especially if using bone marrow graft, there is always a problem with the immune reconstitution and relapse. So we hypothesized that infusion of gamma delta enriched product will boost the immune system and will help also, will decrease the risk of relapse. But instead of trying to selectively infuse a gamma delta product, we decided to infuse an alpha beta T cell depleted product. Because alpha beta T cells are the majority of T cells, like 95% of T cells are alpha beta depleted, and this was the CD4, CD8 is, and these are the main culprit for GVHD. So these products were deployed from alpha beta, and basically they have stem cell, we have gamma delta, they have NK. So it's supposed to be good booster that you give to the patients. But we decided to infuse this product soon after in the post-transplant side and before engraftment. People usually engraft here and we infuse the product soon after finishing the regimen and PT side. So and this what happened. We encountered 2 graft failures in our population. One of them was early reactivation of HHV6. Really bad luck, okay? And the other one was a terrible leukemia. And the patient came with uncontrolled disease. And unfortunately, it failed to engraft because of the disease progression. We perceived this as bad luck, but bottom line, we have to engraft failure. Add to this, we had a patient who developed acute lung injury and died, unfortunately, with acute lung injury. So problem is, these are things that we see often after transplant. We still don't know if this is bad luck or we induced these complications by infusion. For the FDA, it's a no-no, period. You've done this, you've had this, you got to stop. So we stopped here. But we came back and went back to the FDA, and we wrote another protocol from the scratch. But here we used an ex vivo expanded activated gamma delta T cell product. That's a different thing. It's not just alpha beta T cell product, but this is gamma delta selected, and we do in vitro expansion for the gamma delta product. But here, this is a different thing that we've done. We decided to infuse this after engraftment. Okay, so if there is any crap, it's going to happen before engraftment, let it happen, and then we infuse it afterwards. And I want to tell you that the study is going very well. Multiple patients have been enrolled, but unfortunately I cannot share any results right now because it has an industry sponsors and this hasn't been published yet. So in conclusion, or basically the lesson learned from early phase clinical trial and cell therapy, number one, we have to carefully select the design that we're going to use to predict the recommended phase 2 dose, and CRM is definitely better than 3+3 design. We have to be careful enrolling subjects who strictly meet the criteria, so we won't commit the mistake that UPenn committed in 1999 with JC. And the other lesson is careful study planning to avoid overlap with concurrent therapy. I'm here referring to the mistake that we've done with infusing an experimental product before infusion and encountering graft failure. With this, I conclude and happy to answer any question. Thank you.